So where we have more uncertainty then is this, the effect of the long-term ligotrophication. So you can see this is for Lake Michigan, again, through time, the long-term decline in total phosphorus. To what extent is that also contributing all the way up, a couple of different trophic levels up to crayfish? And so our specific charge was to investigate the extent to which these changes in lower trophic levels, so looking at invertebrates, looking at phosphorus, looking at phytoplankton, could be limiting the capacity of crayfish that ultimately support the popular recreational fisheries as well as commercial fisheries. And they asked us to produce a white paper that with very specific components to it, including summarizing the trends for different lower trophic levels, identifying the linkages between lower trophic levels and fish biomass, trying to identify existing knowledge gaps, data gaps, and research priorities. And then finally, are there any indicators that could be reported out at meetings such as this that would give managers a better handle on how these lower trophic levels are affecting fish? So how do we do this? Our process really was done in large part based on conference calls. We had about 5 of them since we started just over a year ago. We had numerous emails, but really the capstone was a workshop that was held in August at Central Michigan University. They have a beautiful biological station at Beaver Island, right in the middle of Lake Michigan. Inspirational setting. We were hosted by Kevin Pangle here and Hunter Carrick here, who are also members of our task team. And we made a lot of progress in those 2 or 3 days on Beaver Island. And then more recently, as this talk is, deadlines are helpful in getting you to move forward on tasks such as this, we began to move quickly via email to try to prioritize the indicators, the monitoring gaps, the knowledge gaps. And I'll share some of those sort of the top 1 or 2 for each of those in this talk. So where are we going here in the next couple of minutes? I'll give not an exhaustive summary of what's going on in lower trophic levels because you've seen that in Mark's talk earlier. But just a few of those, and we'll bring in some other data sets beyond just the EPA that I think are also helpful as we look at especially some of the nearshore data. How can these changes affect fish? Talk about some of those mechanisms that we'll offer as hypotheses for those linkages. Give you the results of our, what are the key indicators, possible indicators, monitoring and knowledge gaps, and then a very early look at the recommendations that we're drafting. Okay, so let's look at some of these lower trophic level trends, and we'll start with phosphorus loading first of all, and there's a couple of things I want you to look at for this particular graph. So we start back in the early 1970s, and the first thing you know is there was a lot more phosphorus loading back in the 1970s, a tremendous drop. So this is the Great Lakes Water Quality Agreement. And then we essentially have pretty much stable loading going on about through 2008, 25 years of stable loading. The other thing to note, however, is that we don't have any loading data for the past 10 years. Is that important or not? Maybe it's not important given the stability we've had over the previous 25 years. At the same time, at least on the U.S. side, we've been investing heavily in the Great Lakes Restoration Initiative, and one of those particular goals is to reduce phosphorus loading. So one might expect that we might even potentially be seeing some reductions if those restoration efforts are effective, and actually seeing further reductions in phosphorus loading in the lakes. Again, we don't have the data to really know that. Now that phosphorus that loads into the lakes, of course, translates into phosphorus that's measured excellently by EPA. Again, this was Mark showed in his talk. And you get these measures of offshore total phosphorus in the spring is a really nice indicator. But what is interesting is more and more studies begin to look at what should the offshore phosphorus concentrations be, given what the loadings are, given what the retention time of Lake Michigan is, and then making some assumptions about the settling coefficients of phosphorus in the lake Those predictions are very different from what we're actually seeing. Some people are predicting that there's more phosphorus out there than should be, perhaps due to internal loading. Others, and that's some work done by Rick Barbiero, some recent work by Mark Rowe that is in review, says that when you extend this out into 2010 or so, there's actually phosphorus concentrations are lower. And that there could, even when you're accounting for the phosphorus built up or contained in mussels and detritus and in phytoplankton. So there's a lot we don't know about what sets, how we move from here to what actually is measured in phosphorus due to the internal phosphorus dynamics in the lake. Okay, so what about phytoplankton? Most of us are familiar with the phytoplankton story in the offshore, which is the one in the open circles, showing back to 1998. These are data measured through remote sensing by Barry Lesch, a contractor through EPA. You can see this long-term decline in the offshore. But when we look at data from the nearshore, and we can only come up with two time series for this. One, this is the NOAA time series, which covers the entire time period. This is nearshore waters, 15 meters site at Muskegon. There is no decline. This is another site. This is offshore of Milwaukee, perhaps a little bit north of Milwaukee by Harvey Bootsma's lab. It doesn't go back to the 1990 to 1998, but over this time period, where mussels, especially quagga mussels, are having an effect, mostly in the offshore, we don't see a nearshore decline in phytoplankton. Another factor that we talked a lot about, especially in the workshop, was the changes in water clarity. And these are pretty striking data. This is from US EPA monitoring once again, going back to the 1980s, some variation, but in general, what I see is a period since 2004, so this would be the period when quagga mussels really got rolling out in offshore Lake Michigan. The water is now 1.6 times clearer than what it used to be in the previous 25 or 30 years. And we talked about the potential ways that this clearer water could also have biotic implications, and I'll get into that in just a little bit. Zooplankton. Again, Mark showed you a lot more in-depth zooplankton data that I'm going to get into. I'm just showing you here total crustacean zooplankton biomass, just relativized, so we can compare the two EPA data sets. This is in the northern basin and the southern basin, and then comparing it to, again, relativized through the NOAA time series in the offshore site, offshore of Muskegon. 2 out of 3 of those are showing you declines in offshore total zooplankton, but we don't see that in the northern basin. This is offshore. Now again, let's go to the nearshore. Not congruence again. Our 2 data sets for the nearshore. This is one. This is back to the NOAA time series at 15 meters offshore of Muskegon. They are seeing declines since the late 1990s in crustacean zooplankton. But Illinois Natural History Survey, Syracuse Chesney, John Detmers used to work on these data. When you take total crustacean zooplankton biomass, there's no trend in the nearshore and southwestern waters. And we don't have any data in the nearshore in the northern basin. Quagga mussels, this is now data from the southern basin that NOAA collects, started by Tom Nalepa, now kept up by Ashley Elgin. If you follow the bold line here, this is the weighted average across all depths. It looks to be relatively stable in the southern basin for quagga mussels. Diparia, most of us are very familiar with this story, the loss of diparia, a very high energy prey resource for fishes. The only somewhat optimistic tone is that we're still seeing diparia when you go out into waters deeper than 90 meters, whether it's NOAA measuring in the southern basin or whether it's EPA measuring sort of lake-wide. So there are still some diparia out there, you just have to go deep to find them. And the final prey that is important to fish is mysis. 2 time series exist for this. One is the NOAA time series back to 1980s, and that's in black. And it's more of a nonlinear relationship, but if you try to fit a linear trend there, there's no pattern. Through the summer acoustic survey that USGS and Michigan DNR participate in, there is a significant decline in mysis densities, but in many ways, that's driven by a very high value that was estimated in 2005. If you were to remove that, there's probably no trend in mysis in this more recent time series either. So to try to summarize, one of the things we wanted to do was, again, to separate near shore versus offshore. And there probably are some other nuances we could get in there in terms of east side of the lake versus west side of the lake, if we have the data to make those discriminations. But on the right-hand side, you can see that most of what we hear is sort of the offshore story where there are a lot of declines. And I'm just showing you here the invertebrates that fish would be consuming. And again, now we're pulling out some of the different large zooplankton taxa to try to drive home the point. But you're seeing a lot more declines in the offshore, not as many declines in the near shore, but we don't also have as many time series to really evaluate that in the near shore. Okay, so the second part then was to try to say, how can these changes be affecting fish, and what are some hypotheses surrounding that? And so to do that, we really took more of a life history perspective, where we, it's an illustration of an alewife. She produces eggs. Those eggs, some of those eggs hatch out and survive, produce larvae. Some of those larvae survive to become juveniles, and of course those juveniles survive to become adults. And what we're interested in is how can changes in lower trophic levels affect different life history components for a particular fish species. So one hypothesis is that this dramatic increase in water clarity could be further facilitating the top-down predation control that has been existing, that has been important for decades. So the idea being that despite declines in alewife densities, for example, salmon may have been able to maintain strong top-down control because it is easier, essentially, to locate and alewife prey, both adults and juveniles, I should say. So that's one potential hypothesis. The second hypothesis takes into account the reductions in growth rates that we've seen. When we look at growth rates in the 80s compared to growth rates in the 2000s for alewife and for bloater, it's about a 30 to 40% decline depending on the species. So we're just doing simple math and saying if you were to have potentially had the same growth rates in the 2000s that were able to be maintained in the 1980s, you would have much higher levels of fish biomass. But that's obviously not happening. So it's essentially a loss in potential biomass that's potentially driven by the declines in invertebrate prey that are leading to these slow growth rates. The third hypothesis is more of a maternal effects one. So now I'm not thinking about growth rates, but thinking about the condition of the fish, their energetic condition. We know that for many fishes, females that are in better energetic condition, that have higher lipids, that are fattier, can produce more eggs, or can produce higher quality eggs with higher lipid profiles. It could potentially lead to more larvae being produced, and more larvae that survive the juveniles. A fourth hypothesis looks at this transition between the larval stage and the juvenile stage, and notes these declines in zooplankton prey, and says that this particular transition could be very vulnerable to the changes in the zooplankton community. And these changes could be not only declines in densities, but it also could be changes in the vertical distribution of zooplankton in the water column. Again, thinking back to that changing water clarity, could be driving zooplankton deeper into deeper waters. Pithetrephes, we know, drive zooplankton down into deeper waters, and whether or not larvae are moving down into deeper waters to overlap with the zooplankton that are there is something that really probably needs further exploration. In the white paper, we talk about the relative evidence we have for each of these four hypotheses, but I don't have time to go into that today. So next, I want to summarize the indicators, the monitoring and knowledge gaps that we were asked to come up with. I'll start with the indicators, and we came up with 16 of them, and every member on the committee voted 1 to 16. What do you think is the best indicator? And what came out on top was reporting out some measure of alewife condition or another prey fish condition, with the idea that the condition or the weight of the fish at a given length, which is what we're showing you right here, would be some way of integrating what prey is available to that particular fish. And so when you take this back into the 1960s, and these are USGS data from our bottom trawl caught fish, you see that, interestingly, as the alewife population declined in size, they did see some they did tend to weigh more at a given length, at a standardized length, through the 1990s or so. And then as Chuck Medengian has published, there was a dramatic drop around 1994, 1995 in alewife size per given length. He also showed this for energetic condition. And since then, it's been either stable or perhaps even somewhat of an increase. Chuck has provided evidence that this is probably due to the loss of diaperia, which was an important part of alewife diets in some parts of the year. And again, that diaperia is a high in prey. The second potential indicator is one you've seen before, just again in Mark Tufton's talk. This is now the Lake Michigan integrated satellite derived chlorophyll estimates. So these are year 1998 through 2016. 98 is the first year we have good satellite information that we can do this with. And then you can look at it from March to November, sort of the ice-free parts of the year, across the entire part of Lake Michigan, not just the few times that we're out there with the boat. And so what you can see is that we've lost essentially the spring bloom that used to occur in Lake Michigan, showing up here in the 80s and 90s. Now we're getting this more fall-oriented bloom. And this is the same thing that Mark talked about in Lake Huron. The other thing you see is there are some years, like 2010 to 2012, where essentially it was low productivity really in all times of the year. Moving now into monitoring gaps, we had 11 different ones that we talked about. The top one actually was, we recognize that there is a need for more coordinated monitoring of the near shore. And I'll talk more about that, specific details in that in the recommendations. We just felt like there wasn't enough information about what's happening in Lake Michigan in the near shore. And ideally that would include larval fish to try to get at some of that potential, that hypothesis of changing zooplankton community affecting larval fish. The second one was the reminder that we needed to continue to make direct measurements of primary production, especially given we now have a lot more indirect measures of primary production out there from satellites, etc. We need to have some way to calibrate that. And the third one here is that we would love to see an update to tributary loading, so that we can see has phosphorus loading changed at all in the last 10 years. And as was done previously by Dave Dolan and Steve Chapra, doing that by tributary would also give you a much better sense of how that loading could be affecting productivity in the very near shore waters. Finally, the knowledge gaps. So the idea here is, what do we know? Obviously there's a lot we don't understand, as Tom Stewart was just saying. We don't want that to paralyze us, but if we could sort of identify what we really want to learn more, the first thing that came out on top in terms of limiting our ability to make the linkages between lower trophic levels and fish is, what are the bottlenecks for fish recruitment? In particular, what role might these changing zooplankton distributions and abundances have on the ability of larval fish to survive? The second one is one that might be a little bit surprising. What is the relative importance of limiting nutrients and aquatic muscle filtration on driving declining primary production? I think most of the story we tend to hear is that the reason there isn't as much phytoplankton is because it's all mussels. So we just kind of wave our hands and say it's a mussel story. But there's more and more evidence that it's nutrients as well as mussels that are interacting to have a limiting effect, and that takes you back into sort of the phosphorus loading aspect. So Dave Warner and Barry Lesh published a paper showing the importance of nutrients, mussels, and precipitation in driving primary production. There's a more recent study by Mark Rowe, who is a scientist at the University of Michigan and has colleagues at Bunnell that is doing a model where let me walk you through this. On the left-hand side, these are satellite images of chlorophyll. And so this part of the lake, it would just mean areas where there was cloud cover, so you couldn't get those retrievals. And then he had models, 3 different models, where he allowed there to be mussels, allowed there to be loading, no mussels but loading, and on the far right he had mussels but no loading. And then essentially what you want to do is you want to say which of these 3 look the most like what we're measuring in nature. And this particular model run was in March, a period when mussels should still be able to really have an impact on filtering the entire water column before stratification sets in. And the take-home point is that when you have mussels and loading, you still have this nearshore enhancement in chlorophyll, much like you're seeing here from the satellite retrieval. But when you don't have loading, when you just have mussels out there, you essentially get no nearshore productivity, which makes sense, I think, from an intuitive standpoint. Another way that he showed that is the summary slide. So these are all the different scenarios Mark ran. And the take-home message is that the model was most sensitive to the different scenarios of loading, less sensitive to the different scenarios of mussels. So again, reinforcing the idea that we don't maybe have as good of an understanding as we think about what's really driving phytoplankton out there in the lakes, phosphorus and the loading of phosphorus, and the internal cycling of phosphorus could be a lot more important or as important as the mussels that are out there feeding on the phytoplankton during 6 months out of the year. Okay, so recommendations, these are very early, and this is something that will take more feedback, hopefully, from the Lake Committee and sort of whether or not we're hitting the kinds of recommendations that they're interested in. But what I can summarize so far is, of course, going back to what Tom just said, scientists always want to do more research, but they did ask us for research priorities. And what we hope to do is that we can use some of the priorities we identified to help encourage funding to try to better understand these linkages. So again, as an example, what's more important, nutrients or mussels? And the answer to that question could lead you to different management strategies. If we find that limiting nutrients are more important, that might get fishery managers a little bit more interested in setting phosphorus target limits, for example. If we find that mussels are more important, there is a very early research into the potential for mussel control, probably not possible at a lake-wide level. But if mussels were to be found to be the key driver, as many of us thought beforehand, then that would be supportive of trying to, and you wanted to change the trajectory of oligotrophication. Mussel control may be an area to invest in research. The second point to make is that as we gain improvements in understanding, that can allow us to do better and to build more realistic scenario-based models. So like the one I showed you that Mark Rowe did. We have other models. Yuchun Cow in our lab has an ecosystem model that's exploring how different nutrient loading scenarios, how different salmon stocking scenarios, and how different dry-seeded mussel densities can affect fish productivity. Ed Rutherford, sitting right here, also has a suite of ecosystem-based models that explores different scenarios. And these models are only as good as the data and the understanding that we have that can be put into these. So that's helpful. But the other part is just that as we gain a better understanding of these linkages, we can use that information, or managers can use that information to inform stakeholders and where needed, modify expectations. The second recommendation is to try to implement a coordinated and standardized nearshore monitoring program to complement the stories and the work we're used to hearing about the offshore already. We think this is important because the nearshore is so dynamic. We know that it's a key spawning and nursery area for many of our important fish species. And so one potential way to go about this is to convene the different groups that are working in the offshore currently, that probably are using to some extent different methods to sample lower trophic levels, try to come up with a standardized approach. And so this would be NOAA, for example, off of Muskegon, Illinois Natural History Survey, Harvey Bootsma's lab, and now Central Michigan is now starting a new nearshore monitoring program as well. So get those groups together that are already working, figure out a way where they can be more standardized, and that standardized protocol could then be shared with agencies, tribes, universities that are interested in expanding the coverage that we have in the nearshore monitoring arena. So with that, that's my last slide. I'd be happy to take any questions if there's time.